Welcome to the Burden Center in Baton Rouge. I'm Chris Clark. I'm a plant pathologist who works on sweet potato diseases. And the reason we're here today is that many of you who are familiar with the LSU Ag Center Sweet Potato Research Program know all about the Sweet Potato Research Station in Chase. That's where we produce clean seed, we do breeding research, and also cultural practices research. But because I work with diseases, we don't want those at that station. So the Burden Center is very important because it provides us a place right here in the heart of Baton Rouge where we can work on diseases. It's right close to campus, so we can get here and do a lot of sampling. And so today we're going to show you a few of the things that are going on in the field this year. Last year, Kathy DeRobertis and I did a little video for the virtual field day, and in that we explained how we produce clean plants in the lab, going through the tissue culture process and the virus testing process. Today I'm going to follow up and talk about some of the things that happen after that stage of development. So we talked last year about being members of the National Clean Plant Network for sweet potato. And one of the things that that group is doing are some economic studies. And they have just started this year, and we're in the plot now where we're doing that. So we have very early generation seed. We call it G1, meaning it's only been in the field for 1 year. And then we have some old generation seed of the same varieties. So here in Louisiana, we're working with Beauregard and Orleans. Each of the other clean plant centers is doing the same thing, but they're using the varieties that are important in their location. The whole idea is that we're going to grow these out and do our best to accurately determine the yield and the quality that are produced from the G1 seed as compared to the old seed. So obviously the old seed we're using because it's more likely to be reinfected with viruses. So here you see the plot. You won't see very many symptoms right now, and that's not unusual for sweet potatoes infected with viruses. Quite often they show viruses only at certain times. So the old generation seed, all the plants we planted were infected. And those plants tend to show what we call chronic symptoms. And you'll see in the overlay slide that those consist of yellow spots that quickly develop purple borders around them. They're usually only on the older leaves. They only last for a little while, and then those symptoms disappear. And that's the stage we're at here. Most of those symptoms have disappeared. The plants have recovered, but they're still infected, and they'll still be affected by the viruses. The new generation plants, in the case of Louisiana, we tested them and found that we didn't detect any virus infection in the G1 seed. And those plants, when they're planted in the field close to the infected plants, will get reinfected, and they'll show a different type of symptom called acute symptoms. And those develop on more leaves. They're a little more pronounced. They're easier to see. And it's something that we hope in the future we can use to help rogue some of the seed and keep it cleaner. So we've already gotten some results from this economic study from the National Clean Plant Network. And you're going to see that in this slide. The older generation seed all has a lot of reinfection. Virtually every plant is reinfected with at least one virus. Some of them have 2 viruses, 3 viruses, or even 4 viruses in them. The G1 seed, in Louisiana, we've been working for 10 years to try to clean up the foundation seed, prevent it from being reinfected at the sweet potato station in Chase. So they do a lot of work to try to manage morning glories. We don't plant anything there that could possibly be infected. So anything that was exposed is not planted there. The other states haven't started doing that yet. And so you're going to see with the G1 seed in this slide that the Louisiana G1 seed was clean, but in other states they already had some reinfection in that seed. So these are plots of about 30 plants. At the end of the season, we're going to measure the yield and the quality. The purple plants here are border plants that we put just so that the plants at the end don't produce a lot of jumbos. Each one of the plants competes with each other plant, and so we add one to the end of the plot just to have a competitor there And we use something that's purple like this so we can tell it apart. So I'm going to move over to another section of the field and show you something else we do. It's a real challenge for each of the clean plant centers to produce enough clean seed to furnish their industry every year. Total in the U.S., we use almost 2 billion sweet potato plants a year. So for these 6 centers to produce that many clean plants is almost impossible. So we're trying our best to produce as many as we possibly can with all of the infrastructure we have available to us. Each state does it somewhat differently. So in Louisiana for the last 22 years, we've been using tissue culture to micropropagate plants to increase them, to take to the greenhouses at the Sweet Potato Research Station in Chase in October. And then from October all the way through to May, they take vine cuttings constantly to increase the number of plants that are available. In some of the other states, they maintain the plants in the greenhouse all the time. So the plants in tissue culture rely on the sugar that's in the tissue culture medium to grow, and they're not really forced to photosynthesize like a normal plant. The plants in the greenhouse do photosynthesize, but sometimes they're subjected to stresses in the greenhouse when it gets too hot. So one of the things we're interested in doing is looking at alternative means of producing more clean plants to help fuel an increase in production of plants, in the hopes that in the future we're going to shift and sell less clean seed in the way of storage roots that we traditionally have used, and more in the way of vine cuttings sold directly to growers. So one of the things we have been working with the last year is trying an alternative to tissue culture propagation, which is to use a modification of hydroponics that we can do in the protected lab environment so they won't get reinfected, but it forces those plants to be in a photosynthetic mode, and it might very well change how they perform. So what you see here is a very small test. These are little 10-plant microplots, we call them, in which we're comparing Murasaki, Bayou Bell, and Beauregard plants that were produced from micropropagation and tissue culture to plants that were produced by the modified hydroponics in the lab. And initially, the hydroponic plants seem to grow out a little more vigorously, but there's a lot yet to be determined on this. So in the fall, we'll harvest this, we'll compare the yield, see if there's any difference, and we'll continue this testing for several years before we really reach a conclusion as to whether it's something we're going to use or not. So that's the second plot I wanted to show you. We're going to move to a third plot and talk about one other thing related to clean plant production. Tissue culture has many advantages for propagating sweet potatoes to increase clean plants. One obvious one is that these plants are grown in containers, so they can't get reinfected in the lab. And we have great confidence that when we do that, we can keep them clean. But there's some disadvantages too. So in this slide, you're going to see that 4 different plants from tissue culture. Can you tell, are they the same variety or not? Actually, they're 4 different varieties. They all look very similar in tissue culture because the plants grow very slowly, they don't produce really large leaves, they don't really have time to develop some of the differences. So it's very easy for us to confuse them. And being human beings, sometimes we make mistakes labeling a plant or something like that. So we have to have some way of making sure that what we have in tissue culture is what it's supposed to be. So in this plot, these 2 rows, we've got about 100 different mericlones of sweet potato. That's sometimes we have 2 or 3 mericlones for one variety. That just means they came from a different meristem. And we're growing them out so that we can get a good reading of all of their characteristics. What color leaf they have, what's the shape of the leaf, what's the pattern of veins on the leaf, what's the color of the veins on the leaf. And when it comes time to harvest, we'll dig these up and look at the roots. We'll see what the skin color of the roots is, what the flesh color of it is, and we'll be a little more confident that everything is what it's supposed to be as it's labeled. So you've now seen some of the things we do as a follow-up to our lab work in producing clean plants. And we're shooting this video on July 19th, 2021. 2 days from now, we're going to have a meeting in Mississippi of a new project called Clean Seed that Mississippi State is heading up. It will involve some of the same states cooperating, but now we're going to start looking at some of the things that happen in the seed program after they leave the era, the region in which NCPN controls activities, which is everything up through production of foundation seed. So hopefully we're going to have a research plan and extension plan developed at that meeting that you should be able to learn more about in September when this video is available online, that will deal with some of the issues of keeping that seed clean as it goes through production. We don't rely solely on clean plants to control diseases, but the clean plant system does fit hand in glove with the breeding program we have here, and it's one of the reasons we spend so much time and effort on the clean plant program, is to make sure that as these new varieties become available, they're clean and they can be shipped anywhere where people might want them. So in order to have new varieties, we have to screen for disease resistance. And we work with about 10 different diseases now that we actively try to screen for resistance and improve the resistance in the new varieties that are released. Historically, one of the most important diseases in Louisiana was streptomyces soil rot. And most farmers will know it just simply as soil rot, or in North Carolina they call it pox. This disease was widespread in Louisiana long before I started. Efforts to develop resistant varieties started in about 1910. It wasn't until about 1977 that Jasper was the first variety released from LSU Ag Center that had resistance. And Beauregard, which many of you may be familiar with, was the first variety that was really adopted and planted on a widespread scale that had resistance. But now all of the new varieties have resistance to this disease. And this is one of the places where we screen for resistance. So this disease is a soil-borne disease. It tends to be worse when the soil is dry, when the pH is above 5.2. And so it takes a little bit of work to manage this disease. It didn't occur here at Burden, so in order to have this plot, back in the 1980s, we had to infest it with the pathogen and build it up, and it took about 7 years to get it built up to the point where we could use this for screening for resistance. So now each year we take all the advanced breeding lines in Dr. Labonte's sweet potato breeding program and screen them for resistance here, and we compare them to a resistant standard, which is Beauregard, and a susceptible standard, which is Jewel. And the other thing we do is we have to manage the soil pH and keep it up high enough to get the good disease. And we use half of this field to grow the susceptible Jewel and build up the pathogen, and the other half to run our test. And then next year we'll switch back and forth. The test will be where the susceptible is now, the susceptible will be where the plot is now. So this is just one of the 10 diseases we work with here. There's another plot very similar to this here that's not infested with streptomyces that we're going to grow and harvest roots, and we'll use those for post-harvest disease resistant studies. And we'll look at Rhizopus soft rot, Fusarium root rot, black rot, bacterial soft rot, Java black rot. I think that's the 5. In the greenhouse, we also do screening for resistance to Fusarium wilt and southern root knot nematode, and now in recent years guava root knot nematode. And if you're interested in the outcome of those, you can go to the Sweet Potato Research Station website and look at the descriptions of diseases, and at the bottom is a table such as the one you see here in this slide that shows a summary of all of these ratings. Each one of those little letters represents a lot of work to determine whether these breeding lines are susceptible or resistant to disease. Well, thank you. We're glad you could join us here at Burden for this year's virtual sweet potato disease tour. I hope you've learned something. You can always refer to our websites for more information, and we'll look forward to seeing you again next year.